Welcome. We're glad you have decided to join us here at the Z Files, as we delve into advances in medical science, technology, and artificial intelligence, or AI, and a scenario in which this might have rewritten history. And as curiosity guides us gently towards sleep, we will also examine how ongoing advances could re-engineer the machinery of health and life itself. Part 1. Origins of Sickness. How ills of the past shaped us. Let us begin by clarifying that we are not here to alarm, but to imagine. Before antibiotics, before vaccines, before the germ theory of disease itself, humanity lived in a world where invisible forces could sweep through a city and leave it hollow. Smallpox, plague, tuberculosis. These names carry weight even now, echoes of a time when sickness was not just personal tragedy but collective catastrophe. Deadly diseases shaped empires, redirected history, and wrote themselves into our DNA. Smallpox was perhaps the most intimate of killers. It did not discriminate. The Variola virus turned human skin into a battlefield. The disease announced itself with fever and ache, then erupted in pustules that covered the body, often the face most densely. Survivors bore the scars for life. Many did not survive. In the 18th century alone, smallpox killed an estimated 400 million people worldwide. It killed more people in the 20th century than all the wars of that bloody century combined. The human response was a mixture of desperation and ingenuity. Variolation, an ancient practice from China, India, and the Ottoman Empire, involved taking material from a smallpox pustule and introducing it into a healthy person through a scratch in the skin. The idea was to trigger a milder form of the disease and confer immunity. It often worked, but it also sometimes killed. When Edward Jenner introduced vaccination in 1796 using the related but less dangerous cowpox virus, he transformed folk knowledge into something safer, something systematic. Still, even vaccination took nearly 2 centuries to eradicate smallpox. The last natural case occurred in 1977. Plague occupies a different place in memory. The Black Death, caused by the bacterium Yersinia pestis, arrived in Europe in 1347 and killed perhaps half the population in some regions within 5 years. Yersinia pestis is a tiny rod-shaped bacterium that normally lives in fleas that feed on rodents. But when trade routes bring infected rats into human settlements, when fleas jump to human hosts, the result is catastrophic. The bacterium multiplies in lymph nodes, creating the painful swellings called buboes. If it reaches the bloodstream or lungs, death often follows within days. Medieval physicians blamed bad air, imbalances of humors, divine punishment. Treatments included bloodletting, poultices of crushed emeralds, or holding sweet-smelling flowers to the nose. None of it helped. The plague reshaped Europe, collapsing feudal systems, redistributing wealth, and leaving a cultural scar so deep that we still speak of devastating events as plagues. Tuberculosis worked more slowly but no less thoroughly. Caused by Mycobacterium tuberculosis, a bacterium with a waxy coat that makes it difficult to kill, TB has been with humanity for millennia. The disease spreads through the air when an infected person coughs, and it settles in the lungs, creating cavities and coughing up blood. In the 19th century, TB was responsible for 1 in 4 deaths in Europe and North America. Sanatoriums, rest, fresh air, these were the prescribed treatments, and they did little. Antibiotics, when they arrived in the mid-20th century, finally offered a cure, but even now, TB kills over a million people each year. These three diseases are woven into the fabric of who we are. They influenced where we built cities, how we organized labor, which bloodlines survived. They drove early attempts at public health and the development of modern medicine. They left genetic signatures in our immune systems, adaptations that helped some populations survive but also created vulnerabilities to other conditions. Now imagine something different. Imagine that the tools we have developed in the last few decades, tools that can edit genes with precision, design vaccines in weeks, and model disease spread with artificial intelligence, could somehow be sent backward. Not literally, but conceptually. What if we could take our current understanding of biology and apply it to these ancient scourges at the moment they first emerged? What would change? This is not a fantasy of time travel, but a thought experiment, a way to understand both the power and the limits of modern biotechnology. The question is not whether we could have prevented these diseases with modern tools. In many cases, the answer is almost certainly yes. The deeper question is what that prevention would mean. If smallpox had never decimated indigenous populations in the Americas, would colonization have unfolded differently? If the Black Death had not shattered medieval Europe, would the Renaissance have occurred in the same way? These are uncomfortable questions, but they are necessary ones. We stand at a strange threshold. For the first time in human existence, we possess technologies that can reach into the fundamental machinery of life and change it. CRISPR gene editing, mRNA vaccine platforms, synthetic biology, artificial intelligence that can design molecules or predict protein structures in hours. The diseases that once seemed invincible are now, in many cases, preventable or curable. But that power brings new challenges. How do we decide which diseases to target? How do we ensure access is equitable? How do we guard against misuse? And how do we think clearly about altering systems we only partly understand? This is where our exploration begins. In the parts ahead, we will look closely at the modern toolkit, the technologies that make this thought experiment possible. We will consider specific scenarios for each disease, imagining interventions that could have changed their course. We will examine the ethical and ecological dimensions, the trade-offs and uncertainties. And we will do so calmly, because the goal is not to frighten or to oversimplify, but to understand. Let us take a breath and move forward, into the realm where biology meets engineering, where intention meets consequence. Part 2: Tools of the New Age, CRISPR, mRNA, and synthetic biology. To understand how modern science might rewrite the story of ancient diseases, we first need to understand the tools themselves. They are technologies built on decades of research, on understanding the language of DNA and RNA, on discovering how viruses and bacteria operate at the molecular level. They are powerful, but also limited, prone to error, and often misunderstood. CRISPR stands for clustered, regularly interspaced short palindromic repeats, a natural defense system bacteria use to fight off viruses. Scientists adapted it, pairing a cutting enzyme called Cas9 with a guide RNA that can be programmed to target any specific sequence of DNA. The result is a molecular tool that can find a particular gene in a genome of billions of letters and cut it precisely. Think of it as a search and replace function for genetic text. A single cut in a genome can be like editing a single sentence in a long, tangled story. It can change meaning, but it can also create unexpected gaps. When CRISPR cuts DNA, the cell tries to repair the break, but the repair process is imperfect. Sometimes the gene is simply disabled. Sometimes the cell can be coaxed into inserting a new sequence, rewriting the gene entirely. Edits can happen in unintended places, off-target cuts that change genes you did not mean to touch. But when it works, CRISPR allows changes to biology that would have been unthinkable a generation ago. In the context of infectious disease, CRISPR offers several possibilities. You could engineer a virus to carry a self-destruct mechanism, limiting its ability to spread. You could edit the human genome to make cells resistant to infection. You could modify bacteria in the environment, making them less likely to harbor disease. Each carries risks. Engineering viruses raises the specter of weaponization. Editing human genomes touches on questions of consent across generations. Modifying environmental bacteria could disrupt ecosystems in unpredictable ways. Now consider mRNA vaccines, a technology that became household during the COVID-19 pandemic but was decades in the making. Traditional vaccines expose the immune system to a weakened or killed version of a pathogen. mRNA vaccines take a different route. They deliver genetic instructions, a strand of messenger RNA that tells cells to produce a specific viral protein. The immune system sees this foreign protein, recognizes it as a threat, and mounts a response. When the actual virus arrives, the body is ready. The elegance lies in speed and flexibility. Once you know the genetic sequence of a pathogen, you can design an mRNA vaccine in days. Production does not require culturing the virus, just synthesizing RNA. For rapidly evolving pathogens or novel diseases, this is transformative. But mRNA is fragile, easily broken down, which is why early vaccines required ultra-cold storage and protective coatings. If mRNA vaccines had existed in the 18th century, smallpox might have been contained within years, not centuries. The Variola virus has a stable genome, meaning a vaccine designed once would likely remain effective. Of course, this assumes global coordination and infrastructure that did not exist then and barely exist now. Synthetic biology goes further. It is the discipline of designing and constructing new biological parts, or redesigning existing ones for useful purposes. Imagine building a bacterium from scratch, programming it to produce medicine, or to detect a disease biomarker and respond autonomously. Synthetic biology has already produced microbes that manufacture insulin, artemisinin for malaria treatment, and spider silk proteins. For infectious diseases, synthetic biology offers tools to create entirely new therapies. You could design bacteriophages to target antibiotic-resistant pathogens. You could engineer probiotic bacteria to produce antimicrobial peptids, protecting against infection from within. Each raises questions about containment, ecological impact, and evolutionary pressure. Alongside these biological tools, artificial intelligence has become an accelerant. AI excels at pattern recognition, finding relationships in vast datasets. In drug discovery, AI can screen millions of molecular structures and predict which are likely to bind to a target protein, shortening the search from years to months. In genomics, AI can analyze genetic variation and identify mutations that confer resistance to disease. In epidemiology, AI models can simulate disease spread under different scenarios. But AI is not a shortcut to certainty. Models are only as good as their data, and biological data is noisy, incomplete, and often biased. Predictions depend on assumptions about human behavior that can shift unpredictably. AI is a powerful lens, but it does not eliminate the need for careful experimentation, ethical oversight, or humility about what we do not know. Robotic labs and high-throughput screening complete the modern toolkit. These systems automate experimentation, running thousands of tests in parallel. In the search for a new antibiotic, a robotic system might test 100,000 candidate molecules in a week. Speed matters when a disease is spreading, but automation also introduces distance between scientist and experiment, risking the unexpected observation that might lead to a breakthrough. Together, these tools form a new kind of medicine, one that is predictive, personalized, and increasingly precise. They allow interventions that would have seemed impossible 50 years ago. But biology is complex, evolved over billions of years, layered with redundancies that resist simple engineering. Every intervention has side effects. Every solution creates new selection pressures. It is useful, and a little humbling, to remember that good intentions are not the same as safe outcomes. The same tools that could eliminate a disease could also be used to create one. The same precision that allows beneficial edits also allows harmful ones. This is not an argument against the technology, but a reminder that technology exists in a human context, shaped by who has access, who makes decisions, and what safeguards are in place. For now, take a breath and let the ideas settle. We have laid out the toolkit, the modern instruments of biological engineering. Next, we turn to the diseases themselves and begin to imagine how these tools might have rewritten their stories. Part 3: Smallpox Reimagined, Engineering the End of Variola. Smallpox was, in many ways, an ideal target for eradication. The virus had no animal reservoir, meaning it could only survive by passing from human to human. It produced visible symptoms, making cases relatively easy to identify. And the vaccine worked reliably. These factors allowed the World Health Organization to declare smallpox eradicated in 1980. But imagine if we had possessed our current capabilities when smallpox first emerged, or even just in the early modern period. What might we have done differently? The Variola virus is a large, complex DNA virus. It enters through the respiratory tract, infects cells in the lungs and lymph nodes, then spreads through the bloodstream to the skin. The virus hijacks the cell's machinery, forcing it to produce thousands of viral copies before the cell bursts and dies. With CRISPR, we could intervene at multiple points. We could target the virus itself, disabling genes essential for replication. We could target the human genome, making cells resistant to viral entry. Or both. One approach would be to engineer a genetic kill switch into the Variola virus, a sequence that causes it to self-destruct after limited replications. This would require identifying essential genes, then modifying them to trigger cell death pathways after a threshold. The modified virus could still infect but could not sustain an epidemic. Viruses evolve rapidly, and any kill switch would face selection pressure to mutate into inactivity. Still, if deployed early, such a tool might have prevented smallpox from establishing as a human pathogen at all. Another strategy involves human genome editing, targeting genes that produce proteins the virus needs to enter cells. If those receptors could be modified or removed, the virus would lose its foothold. CRISPR could edit these genes in human embryos, creating individuals genetically immune to smallpox. This is technically feasible, but it crosses into heritable genetic modification, changing the human germline in ways passed to future generations. The ethical weight is enormous. A protein that serves as a viral receptor may also play important roles in normal physiology. Removing it might confer immunity to one disease while creating vulnerability to another. A less invasive approach would focus on somatic gene therapy, editing cells in living individuals rather than embryos. You could imagine editing immune cells to recognize and attack the virus more effectively, or editing lung cells to resist infection. The challenge is delivery. Getting CRISPR machinery into enough cells, in the right tissues, to provide protection is difficult. And unlike a vaccine, somatic gene edits might need repeating as edited cells die and are replaced. mRNA vaccines offer a faster and more scalable solution. An mRNA vaccine for smallpox could be designed as soon as the viral genome was sequenced, a process that with modern tools takes hours. The vaccine would instruct cells to produce a viral protein, and the immune system would learn to recognize and neutralize it. Unlike the traditional smallpox vaccine, which used live vaccinia virus and carried risks, an mRNA vaccine would be safer and easier to manufacture. Beyond vaccines and genetic edits, modern biotechnology offers antiviral drugs designed with precision. AI-driven drug discovery could screen billions of molecular structures to find compounds that inhibit essential viral enzymes. Once a promising candidate is identified, synthetic biology could produce it at scale. Now consider the broader implications. If smallpox had been prevented in the 16th century, the colonization of the Americas would have unfolded very differently. Indigenous populations suffered catastrophic mortality when European diseases arrived. Some estimates suggest smallpox alone killed more than half the native population in some regions, a demographic collapse that facilitated European conquest. Remove smallpox, and you remove one of the key advantages colonizers held. Indigenous societies, stronger in numbers, might have resisted more effectively. In Europe, smallpox shaped royal succession and political stability. Queens and kings died of smallpox, shifting power and alliances. A world without smallpox would be a world where certain individuals lived who did not, where certain decisions were made or not made. There are also ecological considerations. The Variola virus was part of the human virome, the collection of viruses that interact with our species. Eliminating it removes a selection pressure that shaped human immune systems over millennia. Some researchers speculate that genetic variants conferring resistance to smallpox might now confer susceptibility to other conditions. Pathogens are not separate from evolution. They drive it, and our biology is in part a response to them. Finally, There is the question of what we would do with the eradicated pathogen. Smallpox samples remain in 2 high-security laboratories. The debate over destroying these stocks has continued for decades. With modern synthetic biology, the entire Variola genome could be synthesized from publicly available sequence data, making physical stocks arguably less critical but highlighting that eradication is no longer a guarantee of safety. The tools that could have prevented smallpox are the same tools that could recreate it. We move forward now, from the virus that was eradicated to the bacterium that still lurks, waiting for the conditions that allow it to spread. Part 4. Plague Revisited. Targeting Yersinia pestis at every scale. Yersinia pestis is a study in adaptability and opportunism. Unlike Variola, which required human hosts, Yersinia pestis thrives in a complex ecological cycle involving rodents, fleas, and occasionally humans. It is a zoonotic pathogen, originating in animals and spilling over into human populations when conditions align. Plague has not been eradicated, only controlled, and outbreaks still occur in parts of Africa, Asia, and the Americas. The bacterium is relatively simple. Yersinia pestis has a circular chromosome plus several plasmids carrying genes for virulence factors. When a flea infected with Yersinia pestis bites a human, it regurgitates bacteria into the wound. The bacteria travel to the nearest lymph node, where they multiply rapidly, causing the painful swelling of a bubo. If untreated, infection can spread to the bloodstream or lungs. Pneumonic plague is particularly deadly, killing most untreated patients within days. With modern tools, there are multiple intervention points. We could target the bacterium directly, engineering weaknesses into its genome or developing drugs that exploit its biology. We could target the flea vector. We could target the rodent hosts. Or we could target humans through vaccination or genetic modification. Start with the bacterium. CRISPR could be used to edit Yersinia pestis in the wild, disabling virulence genes. This is called gene drive, where genetic modification spreads through a population faster than normal inheritance. You could engineer a plasmid carrying a CRISPR system targeting essential virulence genes. Bacteria carrying the plasmid would spread the modification to others, gradually diluting fully virulent populations. The risks are significant. Altering wild bacterial populations could have unpredictable ecological effects, and the modification might not remain stable. Another approach focuses on developing new antibiotics. Yersinia pestis is currently susceptible to several antibiotics, but resistance has been observed. AI-driven drug discovery could identify novel compounds targeting bacterial enzymes or structures that are essential and difficult to evolve resistance against. High-throughput screening could test thousands of candidates, compressing the process from a decade to a few years. Vaccines represent a more proactive strategy. An mRNA vaccine for plague could encode proteins from the bacterial surface, such as the F1 capsule antigen. The vaccine could be designed rapidly and manufactured at scale. Animal studies show such vaccines can protect against plague, but human trials have been limited due to low incidence. Now consider interventions beyond the human host. Gene drive technology in fleas could engineer them to be resistant to Yersinia pestis or unable to transmit it. This has been explored for mosquitoes in malaria and dengue contexts. The biology is more challenging with fleas, and there are many species, each with different host preferences. There is also risk of disrupting ecosystems that depend on fleas as a food source. Rodents present another intervention point. With CRISPR, it might be possible to engineer resistance into wild rodent populations, editing genes involved in immune response. This is feasible in laboratory settings but raises profound ethical and ecological questions in the wild. Rodents play crucial roles in many ecosystems. Altering their genetics on a large scale could disrupt these roles in ways that cascade through the food web. A less invasive approach might focus on ecological management, using AI and remote sensing to predict and prevent outbreaks. Machine learning models can analyze environmental data to identify conditions favoring plague transmission. Early warning systems could alert officials to high-risk areas, allowing targeted interventions. Historical context deepens the thought experiment. The Black Death reshaped medieval Europe beyond mortality. Labor shortages weakened the feudal system. Economic shifts contributed to the decline of serfdom and the rise of wage-based economy. If modern biotechnology had prevented the Black Death, the social and economic structures of medieval Europe might have persisted longer, delaying transitions that led to the Renaissance and the modern world. Finally, consider the dual-use problem. The same tools that could disarm Yersinia pestis could weaponize it. CRISPR could make the bacterium resistant to all known antibiotics, or enhance its virulence. This is perhaps the most unsettling aspect of modern biotechnology. The power to heal is inseparable from the power to harm, and the safeguards are social and political, not technical. We have explored plague from the molecular to the ecological. Each intervention offers promise and carries risk. The lesson is that action requires care, foresight, and humility. Let us breathe and prepare to move to the third disease, the slow killer that still takes 1 million lives each year. Part 5. Tuberculosis transformed, disrupting the persistent pathogen. Tuberculosis is ancient, persistent, and maddeningly difficult to eliminate. Unlike smallpox or plague, tuberculosis works slowly, often taking years to progress from infection to disease. Mycobacterium tuberculosis has evolved mechanisms to survive inside macrophages, the very cells meant to destroy it, hiding and entering dormant states that can last decades. A person can carry the bacterium for a lifetime without becoming sick, yet remain capable of transmitting it if infection reactivates. Tuberculosis has likely killed more humans than any other single infectious disease in history. Even now, TB kills over 1 million people annually. Another 10 million fall ill each year, and perhaps 1/4 of the global population carries latent infection. Drug-resistant strains are spreading. Multi-drug-resistant TB requires treatment regimens lasting 18 months or longer, with success rates around 50% in some settings. Extensively drug-resistant TB leaves some patients with no effective treatment. Why is tuberculosis so difficult to treat? The bacterium's waxy cell wall makes it impermeable to many drugs. Its slow growth rate means antibiotics targeting rapidly dividing cells are less effective. Its ability to enter dormancy allows it to survive antibiotic assault. And its tendency to form granulomas, dense clusters of infected cells, creates microenvironments where drug concentrations are low. Patients who feel better after a few months often stop medications, allowing resistant bacteria to survive. Modern biotechnology offers several strategies. The first is to design drugs targeting unique features of Mycobacterium tuberculosis. The bacterium relies on specific metabolic pathways to synthesize its cell wall, to acquire iron, to survive in low oxygen environments. AI-driven drug discovery can screen vast chemical libraries for molecules that disrupt these pathways. One promising target is the enzyme INA, involved in mycolic acid synthesis. The drug isoniazid works by inhibiting INA, but resistance is common. New compounds that inhibit INA through different mechanisms could restore utility. Another target is ATP synthase, which generates energy. The drug bedaquiline targets ATP synthase with good efficacy, though concerns about cardiac side effects limit its use. High-throughput screening has identified several new candidates now in clinical trials. CRISPR offers a different approach. Instead of targeting the bacterium with drugs, we could target the human genome to enhance natural resistance. Certain genetic variants are associated with susceptibility to tuberculosis. Editing these genes in immune cells, particularly macrophages, could make them better at killing ingested bacteria. This is somatic gene therapy and faces delivery challenges, though advances in viral vectors and nanoparticles are making it more feasible. A related strategy involves host-directed therapies, treatments that modify the immune response rather than attacking the bacterium directly. Tuberculosis exploits the immune system, using inflammation to spread while dampening pathways that would clear infection. Drugs that modulate this balance could improve outcomes. The challenge is specificity. The immune system is finely tuned, and intervening in one pathway often affects others. Vaccines represent perhaps the most important long-term solution. The BCG vaccine provides some protection against severe childhood TB, but its efficacy wanes with age. A new TB vaccine providing strong, durable protection in adults could prevent millions of infections. Several candidates are in development, including mRNA vaccines encoding multiple bacterial antigens. Early trials show strong immune responses, but whether those translate into protection remains to be proven. Another vaccine approach uses viral vectors, harmless viruses engineered to carry TB antigens. The viral vector infects cells and delivers antigens, triggering an immune response that mimics natural infection without causing disease. This stimulates strong cellular immunity, critical for TB protection. Imagine deploying these tools in the 19th century, when tuberculosis was the leading cause of death in Europe and North America. Sanatoriums offered rest and fresh air, interventions that sometimes helped but often did not. An mRNA vaccine might have prevented most infections. Effective antibiotics could have cured active cases in months, preventing transmission. The cumulative effect would be profound, not just in lives saved but in social and economic productivity preserved. But there are complications. Tuberculosis is intimately tied to poverty, overcrowding, malnutrition, and lack of healthcare access. A vaccine or drug is only effective if people have access to it. Historical vaccination campaigns often failed in marginalized communities due to lack of infrastructure or distrust. The wealthy would benefit first, widening health disparities. There is also evolutionary pressure. Every intervention creates selection for bacteria that can evade it. Widespread vaccine use might prevent most infections, but bacteria capable of evading vaccine-induced immunity would have a selective advantage. This does not mean avoiding vaccines, but using them wisely, in combination with other interventions. Ecological and evolutionary considerations extend beyond the bacterium. Mycobacterium tuberculosis is related to environmental mycobacteria living in soil and water. Gene flow between species, though rare, is possible. Interventions targeting Mycobacterium tuberculosis could have unintended effects on related species, potentially disrupting soil ecosystems or creating new opportunistic pathogens. Finally, consider the modern context. Tuberculosis persists not because we lack tools to fight it, but because those tools are unevenly distributed. We have effective antibiotics, but treatment regimens are long and difficult. We have diagnostic tests, but they are often unavailable in rural settings. The primary barriers are not scientific but social, political, and economic. Technology alone cannot solve problems rooted in inequality. What we have imagined for tuberculosis is a suite of interventions, each addressing a different aspect. Together, they could dramatically reduce TB burden, perhaps even eliminate it as a major threat. But they would not do so automatically. They would require sustained effort, global cooperation, and commitment to equity. Let us pause and gather ourselves before moving forward. We have explored 3 diseases, each with its own biology, history, and potential for intervention. Next, we turn to broader implications, the ethical and ecological questions that arise when we wield such power. Part 6: Consequences and Considerations, Ethics, Ecology, and the Limits of Intervention. Every intervention has consequences, some intended, many not. Biology is not a machine that responds predictably. It is a web of interactions, resilient in some ways and fragile in others. When we intervene, we do so with incomplete knowledge, and outcomes cascade in directions we cannot always foresee. Start with the most immediate ethical question: who decides? The technologies we have discussed are concentrated in wealthy countries, in well-funded laboratories. If a decision is made to deploy a gene drive against Yersinia pestis, who makes that decision? Communities most affected by plague often have the least voice in global decision-making. A gene drive released in one region does not respect borders. It spreads, affecting people who may not have consented or been consulted. This requires frameworks for consent, representation, and accountability that do not currently exist at necessary scale. Consider also access. A new mRNA vaccine for tuberculosis would likely be expensive initially. The cost would decline over time, but in the interim, the vaccine would be available primarily to those who can afford it. This pattern has played out with HIV antiretrovirals, hepatitis C drugs, and COVID-19 vaccines. Life-saving tools sit unused while poorer populations wait. Infectious diseases do not respect wealth or borders. Leaving populations unvaccinated creates reservoirs where pathogens evolve and spread. Equity is not only a moral imperative but a practical necessity. Then there is the question of unintended consequences in human biology. Imagine we engineer genetic resistance to smallpox, editing genes that produce viral receptors. Those receptors likely have other functions we do not fully understand. Removing them might protect against one disease while creating vulnerability to another. The human genome is not a simple instruction manual. It is a network of interdependent elements, where changes in one area can propagate unpredictably. This is especially concerning with heritable genetic modifications, changes made to embryos passed to future generations. Once a genetic edit enters the germline, it becomes part of the human gene pool, and reversing it is extraordinarily difficult. If the edit has unforeseen negative effects, those effects persist and spread. Ecological consequences are equally complex. Every organism exists in a web of relationships. When we remove or alter one element, the web shifts. If we eliminate Yersinia pestis from wild rodent populations using a gene drive, rodents no longer subject to plague-induced die-offs might increase in number, putting pressure on food resources, potentially displacing other species. Predators might thrive initially, then face starvation if rodent populations crash. Pathogens themselves are part of ecosystems. They regulate population sizes, drive evolutionary adaptations, and interact with other microbes. Broad-spectrum interventions could disrupt microbial communities, with consequences for human health that are difficult to predict. We are only beginning to understand the microbiome's role in digestion, immunity, mental health, and chronic disease. There is also risk of evolutionary arms races. Pathogens evolve rapidly. Any intervention creates selection pressure for resistance. We have seen this with antibiotics, where overuse has led to resistant strains that render entire classes of drugs ineffective. The same could happen with gene drives, CRISPR therapies, or any tool that exerts strong selective pressure. Now consider the dual-use problem. CRISPR can edit genomes to cure disease or create weapons. Synthetic biology can produce medicines or engineer pathogens with enhanced virulence. AI can design drugs or toxins. The knowledge and techniques are the same. The difference lies only in intent. This democratization of technology has benefits, but it also lowers barriers to misuse. Preventing misuse requires technical safeguards, regulatory oversight, and international cooperation. Technical safeguards might include kill switches preventing engineered organisms from surviving outside controlled environments. Regulatory oversight involves laws criminalizing harmful uses and monitoring high-risk activities. International cooperation is necessary because biology does not respect borders. Ethical frameworks also need to evolve. Traditional medical ethics apply to individual patients and decisions. But interventions like gene drives or global vaccination campaigns operate at population or ecosystem level, where stakes and trade-offs are different. How do we balance individual autonomy with collective good? How do we weigh benefits of preventing disease against risks of ecological disruption? How do we ensure justice when costs and benefits are distributed unequally? There is also the question of what we owe the future. Decisions we make about biotechnology shape the world future generations inherit. If we edit the human germline, those changes persist. If we release a gene drive, it spreads and evolves. Future generations will live with consequences of our choices but have no voice in making them. This suggests need for caution, for reversible interventions where possible, and for humility about our ability to predict long-term outcomes. Consider also opportunity costs. Resources devoted to developing CRISPR therapies are resources not spent on improving sanitation, addressing malnutrition, or combating climate change. These basic interventions save lives and prevent disease at scale. The allure of cutting-edge biotechnology should not distract from foundational public health measures. Finally, there is the cultural and psychological dimension. How we relate to disease shapes how we relate to life, death, and suffering. In a world where ancient diseases are eliminated, mortality becomes less visible. This could foster a sense of invincibility, that technology can solve all problems. Such beliefs can lead to overconfidence, to reckless interventions that ignore complexity. Alternatively, elimination of major diseases could free human attention for other challenges. What emerges from these considerations is a picture of biotechnology as powerful but not omnipotent, beneficial but not without risk. The tools can rewrite disease history, but they cannot write a simple ending. Every intervention opens new chapters, new challenges, new questions. We have covered much ground. In the parts ahead, we will explore what other diseases might emerge in a world without the ancient killers, how artificial intelligence amplifies these challenges, and what principles might guide us forward. Part 7. The Ecology of Absence. What fills the void when plagues vanish? There is a principle in ecology that nature abhors a vacuum. When a species disappears, when a disease is eliminated, the space it occupied does not remain empty. Other organisms expand to fill it, sometimes predictably, often in ways that surprise. Remove ancient killers, and something shifts. Imagine a world where smallpox never became endemic in human populations. The virus likely jumped to humans from an animal host thousands of years ago when agriculture brought people into closer contact with animals. If that spillover had been contained, smallpox might have remained a rare zoonotic infection. In such a world, the selective pressure that favored certain immune system variants would be absent. Populations that historically developed genetic resistance might carry different profiles, with different susceptibilities and strengths. The absence of smallpox would also remove a major driver of vaccination infrastructure. The success of Jenner's vaccine and the eradication campaign demonstrated that coordinated international effort could eliminate a disease. It provided a template for later campaigns against polio and measles. Without that proof of concept, would societies have invested as heavily in vaccination programs? Other diseases might have driven similar developments, but the timeline would differ. Consider demographic effects. Smallpox killed preferentially among the young but also adults who had never been exposed. Remove smallpox, and you preserve millions of lives, particularly young lives. This changes population growth rates, family structures, economic productivity. But populations do not simply grow unchecked. Other limiting factors exist: food scarcity, other diseases, violence. In smallpox's absence, these other factors might become more prominent. Now turn to plague. Yersinia pestis exists in nature as a rodent pathogen. If we successfully eliminated it from wild reservoirs using gene drives, we would alter the ecology of rodent populations, of fleas, of predators, and scavengers. Rodents that previously experienced periodic plague-induced die-offs might stabilize at higher densities or fluctuate for different reasons. The ripple effects could extend to vegetation and soil health. Other pathogens might also respond. Rodents carry many diseases, not just plague. Hantaviruses, leptospirosis, salmonella, various parasites. In plague's absence, which periodically reduces rodent populations, these other pathogens might find more hosts, greater opportunities to spill over into humans. The net effect on human health might still be positive if plague was the deadliest, but it might be neutral or negative if other diseases increase. The social and economic consequences are equally complex. The Black Death reshaped medieval Europe by disrupting feudal labor systems. In a world where plague did not occur, those systems might have persisted, delaying economic and social changes that led to capitalism, democracy, and industrialization. Or different pressures might have driven similar changes through different pathways. Tuberculosis presents a different scenario. It is a chronic disease embedded in human ecology for millennia. Eliminating tuberculosis as a human disease would not remove mycobacteria from the environment, but it would remove a significant selective pressure on human immune systems. Genetic variants conferring resistance to TB might become less common over generations, potentially changing the landscape of immune-related disorders. There is also the question of what diseases might emerge or become more prominent. Disease ecology is competitive. Pathogens compete for hosts, for transmission opportunities, for ecological niches. When one pathogen is removed, others may expand. HIV, Ebola, SARS, COVID-19. These are zoonotic diseases that emerged in recent decades. Their emergence is driven by deforestation, urbanization, international travel, factors that would exist regardless of whether smallpox, plague, or tuberculosis were present. We must also consider evolutionary pressures on pathogens. Diseases evolve in response to hosts and medical interventions. Antibiotics drive resistance evolution. Vaccines drive immune escape variants. In a world where we aggressively deploy biotechnology against infectious diseases, pathogens that survive are those that can adapt quickly. We might inadvertently select for diseases that are harder to treat, more variable, more adept at evading interventions. Consider antibiotic use as an example. Antibiotics have saved countless lives, but widespread use has driven evolution of resistant bacteria. We now face a post-antibiotic future where common infections could again become deadly. The void created by effective antibiotics is being filled by resistant strains. A similar pattern could emerge with any powerful biotechnology. This leads to an uncomfortable realization. Some level of infectious disease may be an inescapable part of the human condition, not because we lack tools to fight it, but because the fight itself creates new challenges. Pathogens evolve, ecosystems shift, interventions have unintended consequences. This does not mean accepting preventable suffering. It means approaching efforts with realistic expectations and strategies that are adaptive. There are also diseases we have not discussed that might become more prominent. Fungal infections are increasing, particularly drug-resistant strains. Parasitic diseases affect hundreds of millions but receive less attention. Chronic diseases like heart disease and cancer are now leading causes of death globally, and while not infectious in the traditional sense, some have infectious components. The ecology of absence is not just biological but social and cultural. Collective memory of epidemic disease shapes how societies respond to new threats. Communities that have experienced devastating outbreaks maintain vigilance. Communities that have not may become complacent, defunding public health systems. The absence of ancient plagues could breed dangerous complacency. On the other hand, absence of ancient diseases could free mental and emotional space for other concerns. Societies not burdened by constant epidemic threat might invest more in education, arts, and scientific inquiry. The Renaissance, some argue, was facilitated in part by demographic and economic changes following the Black Death. What fills the void when plagues vanish is not a simple answer. It is a cascade of changes, biological and social, predictable and surprising. Some voids are filled by other diseases, other challenges. Some are filled by human choices about resource allocation. And some remain unfilled, spaces where absence itself becomes a presence. The lesson is humility. We can imagine interventions, predict some consequences, but the full scope extends beyond our sight. Let us carry this awareness forward as we consider the role of artificial intelligence in accelerating these changes. Part 8. Intelligence Augmented. How AI reshapes the biological frontier. Artificial intelligence has become inseparable from modern biotechnology. AI amplifies human insight, allowing researchers to explore vast possibility spaces, identify patterns in datasets too large for manual analysis, and optimize designs through thousands of iterations in hours. This acceleration is both exhilarating and unsettling. Start with drug discovery. Traditionally, finding a new drug was trial and error. Chemists synthesized compounds, tested them, observed results, and iteratively refined designs. This took years and cost billions, with high failure rates. AI changes this by predicting which compounds are most likely to work before synthesis. Machine learning models, trained on databases of chemical structures and biological activities, can evaluate millions of potential drugs in silico, scoring them for binding affinity, solubility, toxicity, and membrane permeability. These predictions are not perfect. A molecule scoring well in a computer model may fail in a living organism. Biology is messy, but AI narrows the search space, allowing researchers to focus experimental effort on promising candidates. In tuberculosis, AI has identified compounds inhibiting Mycobacterium tuberculosis through novel mechanisms. Some are now in preclinical testing. Protein structure prediction is another area of dramatic progress. Proteins perform most cellular functions, and their function depends on three-dimensional structure. For decades, determining protein structure required techniques like X-ray crystallography, processes taking months or years. AI systems like AlphaFold can now predict protein structures from amino acid sequences in hours. This opens possibilities for understanding how pathogens work and how to disrupt them. Knowing the structure of key bacterial proteins allows researchers to design molecules that fit precisely into active sites, blocking function. This structure-based drug design is faster and more rational. The limitation is that structure alone does not tell the whole story. Proteins are dynamic, changing shape in response to their environment. AI also plays a critical role in epidemiology and disease surveillance. Predicting how disease will spread requires modeling complex systems with many variables. Machine learning models can integrate data from diverse sources: satellite imagery showing environmental changes, social media indicating symptom clusters, genomic sequences revealing pathogen evolution. During COVID-19, AI models forecasted case numbers, identified high-risk regions, and evaluated intervention impacts. These models are only as good as their data. Surveillance systems are stronger in wealthy countries, creating blind spots. Social media data skews young and urban. Genomic sequencing requires infrastructure not universally available. AI can help fill gaps by making inferences from limited data, but it cannot create information that does not exist. Another application is in designing synthetic organisms. Genetic circuits, combinations of genes performing specific functions, can be extraordinarily complex. Designing circuits that work reliably is difficult because biological parts do not always behave as expected when combined. AI can simulate how different genetic elements will interact, optimizing designs before they are built. The speed AI enables is a double-edged sword. It allows rapid response to emerging threats. A new pathogen can be sequenced, proteins modeled, potential drugs or vaccines designed, all within days. This was demonstrated during COVID-19. But speed reduces time for reflection, for considering ethical implications, for consulting affected communities. Decisions that would have taken years can now be made in weeks. This compression can lead to hasty choices. There is also the dual-use problem, amplified by AI. The same algorithms that design beneficial drugs can design harmful ones. The same models predicting disease spread can optimize bioweapon release. The same synthetic biology tools creating therapeutic organisms can create dangerous pathogens. AI makes these processes faster and more accessible. Governance struggles to keep pace. Regulations designed for slower models are ill-suited to a world where AI accelerates innovation. International agreements lack enforcement mechanisms for monitoring AI-assisted research. Some researchers have called for self-regulation, voluntary agreements to avoid high-risk experiments. This works only if most comply, and history suggests voluntary measures are fragile. AI also raises questions about interpretability and trust. Many modern AI systems operate as black boxes. They take inputs, process them through layers of mathematical transformations, and produce outputs, but internal logic is often opaque. A model might predict a molecule will bind to a target protein but not explain why. This makes it difficult to know when to trust a prediction. Efforts are underway to make AI more interpretable, to design models that can explain their reasoning. This is particularly important in medicine, where decisions have life or death consequences. If an AI-designed drug causes unexpected side effects, investigators need to understand why the model predicted it would be safe. Consider also bias in AI systems. Models learn from data, and if data reflects historical biases, the model reproduces them. In medical research, most data comes from populations that are white, male, and from high-income countries. An AI model trained on biased data will perform poorly when applied to underrepresented populations. There is a broader question about what it means to augment human intelligence with artificial intelligence. Are we enhancing our capabilities or offloading cognitive work to systems we do not fully understand, creating dependencies that could be dangerous if systems fail? The answer is probably both. Used wisely, AI can accelerate progress. Used carelessly, it can amplify errors. In the context of rewriting disease history, AI is the accelerant making many interventions feasible on practical timescales. Designing a CRISPR edit, optimizing an mRNA vaccine, predicting how a gene drive will spread, modeling ecological consequences, all benefit from computational power. But AI does not make the decisions. Humans do. We choose which diseases to target, which interventions to pursue, which risks to accept. As we move forward, the integration of AI into biotechnology will deepen. Models will become more accurate, more interpretable, more accessible. The timeline from idea to intervention will continue to compress. This creates urgency around our questions. How do we ensure equitable access? How do we govern dual-use technologies? How do we maintain humility in the face of accelerating capability? The intelligence we need most is not artificial but moral. The capacity to imagine not just what we can do, but what we should do. Let us take a breath and prepare for the next stage, where we consider what new challenges might emerge and what principles might guide us. Part 9. New Frontiers, New Afflictions. What emerges in a reshaped world? Disease does not simply vanish when one pathogen is defeated. The human condition includes vulnerability, and where one threat recedes, others emerge or intensify. Biology is dynamic, and the relationship between hosts and pathogens is one of constant adaptation. Start with pathogen spillover. Most emerging infectious diseases in recent history have been zoonotic. HIV came from primates. Ebola resides in fruit bats. SARS, MERS, and COVID-19 originated in animals before adapting to human transmission. These spillovers are driven by human behavior. Deforestation bringing people into contact with wildlife, agricultural practices creating interfaces between domestic and wild animals, urbanization concentrating populations, international travel spreading diseases globally. In a world where resources are freed from fighting smallpox, plague, and tuberculosis, we might redirect them toward preventing spillovers. Surveillance systems could monitor wildlife populations for novel pathogens. AI could analyze genomic data to predict which pathogens are likely to adapt to human hosts. Habitat preservation could reduce human-wildlife contact. These might reduce spillover frequency but would not eliminate them. Moreover, some spillovers are facilitated by our interventions. Intensive agriculture creates ideal conditions for pathogens to evolve and amplify. Avian influenza and swine flu have emerged through industrial farming. Antibiotic use in livestock has driven resistant bacteria evolution. Addressing these requires rethinking agricultural practices, not simple biotechnological fixes. Consider also diseases with infectious components. A significant fraction of cancers are caused or influenced by infections. Human papillomavirus causes cervical cancer, hepatitis B and C cause liver cancer, Helicobacter pylori contributes to stomach cancer. Vaccines exist for some, and antiviral drugs can cure hepatitis C. If deployed universally, a substantial cancer burden could be prevented. Chronic diseases, heart disease, diabetes, chronic respiratory conditions, are now leading causes of death globally. These are influenced by genetics but also by lifestyle, environment, and social determinants. Addressing them requires different tools than for infectious diseases. Public health campaigns, food system changes, urban planning encouraging physical activity, pollution reduction. Biotechnology has a role through precision medicine and gene therapies, but primary levers are social and political. There is also possibility of entirely new diseases, pathogens that do not yet exist but could emerge through evolution or be created deliberately. Environmental change creates stress on ecosystems and drives adaptation. Pathogens previously confined to certain regions might expand their ranges as temperatures rise. Vector-borne diseases like malaria and dengue are expanding into regions previously too cold. Fungal pathogens are evolving thermotolerance, with species like Candida auris emerging as serious threats. Synthetic biology raises the specter of designer pathogens. A lab working on a defensive vaccine might accidentally or intentionally engineer a virus with enhanced transmissibility or lethality. A non-state actor might use publicly available tools to create a bioweapon. The barriers are lowering as technology becomes cheaper and more accessible. Preventing designer pathogens requires multiple layers. Technical measures include monitoring synthesis of dangerous genetic sequences. Regulatory measures include laws criminalizing biological weapons creation. Cultural measures include fostering norms within the scientific community that certain experiments are too dangerous. Even with best safeguards, accidents happen. Laboratory-acquired infections are not uncommon. There is also the question of what happens to human health in the absence of certain infectious pressures. The hygiene hypothesis suggests reduced exposure to infectious agents during childhood can lead to increased allergic and autoimmune diseases. The immune system evolved in an environment rich with parasites and pathogens. In their absence, it may become dysregulated. Some evidence supports this. Autoimmune diseases and allergies are more common in wealthy, industrialized countries. If we eliminate major infectious diseases, we might see an increase in immune-mediated conditions. This would not necessarily be a net negative. These conditions are serious but generally less deadly than infections they might replace. Still, it represents a shift in the disease landscape. Mental health is another frontier that could become more prominent. In a world where fewer people die young from infectious disease, where life expectancy increases and populations age, mental health conditions like depression, anxiety, and dementia might become dominant concerns. Biotechnology is beginning to address mental health, but it is deeply intertwined with social factors: loneliness, economic insecurity, trauma, inequality. There is also the challenge of aging itself. As infectious diseases are controlled and life expectancy increases, age-related conditions become more common: cancer, cardiovascular disease, neurodegeneration, frailty. Biotechnology offers potential interventions, but extending health span requires understanding processes fundamental to biology. What emerges in a world without ancient killers is not a disease-free utopia, but a world with a different set of health challenges. Some are infectious, new zoonoses, resistant pathogens, accidentally or deliberately created organisms. Some are chronic. Some are mental. The tools to address these will continue to evolve, but so will the challenges. This is the paradox of progress. Each solution creates new conditions, new selective pressures, new problems. Recognizing this helps calibrate expectations. Biotechnology can reduce suffering, extend life, improve quality of existence, but it cannot eliminate vulnerability or uncertainty. What we can do is make thoughtful choices about which boundaries to push, how to push them, and what safeguards to put in place. Let us carry these reflections forward as we consider what principles might guide us. Part 10: Guiding Principles, Ethics, Equity, and the Governance of Power. We have explored technological possibilities and their consequences. Now we must ask, how do we decide what to do? Decisions about deploying gene drives, editing human genomes, releasing synthetic organisms, or using AI to design pathogens or cures are not purely technical. They are ethical, political, and deeply human. They require principles and governance. Start with the principle of precaution. When consequences are uncertain, especially when potential harms are large or irreversible, caution is warranted. This does not mean paralysis, but proceeding carefully, gathering evidence, testing assumptions, building in reversibility. A gene drive released cannot easily be recalled. A heritable genetic edit cannot be undone in future generations. But precaution must be balanced against the cost of inaction. Waiting too long can cost lives. The challenge is finding the right balance. Another principle is proportionality. The level of intervention should match problem severity. For smallpox, which killed hundreds of millions, aggressive interventions might be justified. For mild illnesses, less aggressive approaches are appropriate. Proportionality requires considering not just aggregate numbers but distributions, who bears the burden and who benefits. Equity is both a principle and a practical necessity. Infectious diseases do not respect borders. A pathogen emerging in one region can spread globally. An intervention available only to the wealthy leaves reservoirs where pathogens persist and evolve. Equity means ensuring benefits are accessible to all who need them. This requires addressing not just technology cost but infrastructure needed to deploy it. Achieving equity requires confronting structural inequalities. Most research dollars are spent on diseases affecting wealthy populations. Neglected tropical diseases receive a tiny fraction of global health funding. Changing this requires changing incentives, public funding for neglected disease research, patent reforms making treatments affordable, technology transfer allowing local manufacturing. Transparency is essential. Decisions about biotechnology should be made openly, with clear justifications, accessible data, and opportunities for scrutiny. Some aspects involve sensitive information that could be misused, but restriction should be the exception. Transparency builds trust, necessary for public acceptance. Participation and consent are closely related. Those affected by a decision should have a voice in making it. For individual interventions, informed consent is established. But what about interventions affecting communities or ecosystems? A gene drive released in one region might spread to others. Who consents on behalf of those populations? These are difficult questions. What we can do is create processes that are as inclusive as possible. Accountability is the flip side of power. Those wielding biotechnological tools should be answerable for consequences. If a gene drive causes unintended harm, who is responsible? If a lab accident releases a dangerous pathogen, who is accountable? Legal and regulatory frameworks exist but are often inadequate. Strengthening accountability requires international cooperation and updating laws. Reversibility is a principle applying where possible. Interventions that can be reversed are preferable to those that cannot. Somatic gene therapy is more reversible than germline editing. A drug that can be stopped is safer than an intervention that cannot be undone. Not all interventions can be made reversible, but where possible, it should be a design criterion. Adaptive management involves treating interventions as experiments, monitoring outcomes, learning from what happens, and adjusting based on new information. This is particularly important for interventions in complex systems. A gene drive deployed in a small, isolated population can be monitored, and if problems arise, corrective measures can be taken before scaling up. Humility is perhaps most important. It is recognition that we do not know everything, that our models are incomplete, that biology is more complex than our understanding. Humility does not mean inaction, but approaching interventions with respect for what we do not know, with openness to being wrong, and with willingness to change course. Governance structures need to embody these principles. At the international level, agreements provide frameworks but are limited in scope and enforcement. Strengthening them requires political will and resources. New institutions may be needed, independent bodies with authority to evaluate high-risk research. At the national level, regulatory agencies play critical roles but often struggle to keep pace. Gene therapies, synthetic organisms, AI-designed drugs challenge existing categories and require new approaches. At the community level, governance means involving those most affected. Indigenous communities have deep knowledge of local ecosystems and should be partners in decisions about gene drives. Patients should have a voice in research priorities. Communities near laboratories working on dangerous pathogens should have a say in safety protocols. Education and public engagement are essential. Biotechnology is complex. Public engagement should not be one-way but dialogue, where scientists listen to concerns and where non-experts contribute perspectives experts might miss. Part 11. The human element, culture, memory, and what we lose when we win. Disease is not only biological. It is a human experience, woven into culture, memory, and identity. Diseases that have shaped history have also shaped how we tell stories, understand suffering, and relate to mortality. When we imagine eliminating these diseases, we must also imagine what happens to cultural landscapes they helped create. Start with disease in art and literature. Tuberculosis, with its slow wasting, became a romantic trope in the 19th century. Characters in novels suffered beautifully from consumption, their illness conferring tragic nobility. This romanticization was problematic, obscuring reality of suffering and making poverty seem aesthetically appealing. But it was also culturally productive, inspiring works exploring fragility, transience, and tension between body and spirit. Plague left an even deeper mark. The Black Death appears in literature, art, and folklore. The Decameron uses plague as a frame for examining human nature under extreme stress. The Danse Macabre emerged from plague-era reflections on mortality. These cultural artifacts continue to resonate, shaping how we imagine pandemics. Smallpox too has left traces. The practice of variolation and vaccination became a contested site for debates about risk, bodily autonomy, and government authority. Anti-vaccination movements arose almost as soon as vaccination was introduced. The cultural memory of smallpox inoculation informs current attitudes toward vaccines. Disease also shapes social structures and community bonds. Caring for the sick, mourning the dead, organizing collective responses creates social ties and reinforce norms of mutual aid. Communities that have survived epidemics often develop identity rooted in that survival, stories passed down through generations. Remove the epidemic, and you remove part of the foundation on which community identity is built. There is also the question of what we lose when we lose the struggle. Human beings find meaning in overcoming adversity. The narrative of triumph over disease is a powerful cultural force. It shapes how we think about progress, human agency, and the value of perseverance. If diseases are eliminated before they can cause widespread suffering, the opportunity for that triumph is also eliminated. Consider disease's role in shaping moral and philosophical thought. Suffering raises questions about justice, about why bad things happen. Theodicy, reconciling suffering's existence with belief in a benevolent deity, has been central in many religious traditions. Plague challenged notions of divine justice. Tuberculosis became a lens for thinking about fate and free will. Memory itself is transformed by disease. Survivors carry memories of illness, and those memories shape behavior, instill caution, drive empathy, or fear. Communities carry collective memories of epidemics encoded in stories, monuments, place names. These are not just historical events but living memories informing how we respond to new threats. There is a paradox. Eliminating disease removes suffering, which is clearly good. But it also removes cultural, social, and psychological responses to suffering, some of which have value. The challenge is preserving lessons and meaning without preserving suffering itself. This requires intentionality, actively choosing to remember, to tell stories, to honor the struggle even in its absence. Education plays a role here. Teaching history, literature, and ethics that engage with disease reality helps preserve cultural memory and insights it contains. Medical students learn about medicine's history, not just triumphs but failures. This cultivates humility and critical thinking. There is also the question of identity. For some people, disease is part of who they are. Survivors of serious illness often describe it as transformative, changing priorities, relationships, and self-understanding. Disability communities have developed cultures and identities asserting that disability is human diversity with its own value. Grief is another human element disease evokes. Loss of a loved one to disease is profound grief. It has inspired poetry, music, activism, social movements. The AIDS memorial quilt is both monument and tool for advocacy. In a world where fewer people die young from disease, there would be less of this particular grief, which is good. But grief is also part of how we process loss. Empathy is often deepened by shared experience of suffering. People who have been seriously ill often report greater compassion. Societies that have endured collective trauma often develop stronger social safety nets. In a world with less disease, some of this empathy might be harder to cultivate. What we are circling around is recognition that disease, for all its horror, has been a generative force in human culture. It has prompted questions, inspired art, shaped values, created communities. Eliminating disease eliminates the generative force, which is the right thing to do, but it means we must be intentional about preserving what was valuable in the responses. This is the human element, the dimension of experience that technology alone cannot address. Biotechnology can prevent infection, cure disease, extend life. But it cannot by itself create meaning, foster community, or cultivate virtue. Those remain human tasks. Let us take one final breath and turn to the conclusion, where we gather threads and consider what it means to move forward with both power and humility. What emerges is a picture of biotechnology as a collective endeavor embedded in social, political, and ethical contexts. The tools are powerful, but their impact depends on how they are governed, who has access, and what values guide their use. Technology alone does not determine outcomes. Human choices do. We are not at the end. There is more to consider. But we have laid a foundation, a framework for thinking about how to wield power to rewrite disease history without losing sight of what matters most. Part 12. Moving Forward. The Responsibility of Possibility. We have journeyed through history and biology, through technology and ethics, through culture and consequence. We have imagined what it might mean to rewrite the story of ancient diseases, to wield tools that can edit genomes, design vaccines in days, predict outbreaks, and alter ecological foundations of infectious disease. Now we return to the central question. What do we do with this power? The first thing to acknowledge is that we are already moving forward. The technologies we have discussed are present realities. CRISPR is in clinical trials. mRNA vaccines are being distributed globally. AI is accelerating drug discovery. Synthetic biology is producing medicines. The question is not whether to use these tools but how to use them wisely. Wisdom means several things. It means proceeding with evidence, basing decisions on best available science, updating as new information emerges, and acknowledging uncertainty. It means considering consequences beyond the immediate, thinking through ecological effects, evolutionary pressures, social impacts, and cultural dimensions. It means listening to diverse voices, including those most affected. And it means acting with humility, recognizing limits of our knowledge and control. One practical implication is prioritizing interventions that are reversible, targeted, and well understood over those that are irreversible, broad, and speculative. Somatic gene therapy is less risky than germline editing. Vaccines are less risky than gene drives. Drugs that can be stopped are safer than interventions that cannot be undone. This does not mean never pursuing riskier approaches, but doing so only when potential benefit is very high and safeguards are in place. Another implication is investing heavily in infrastructure of safety and oversight. This includes regulatory agencies with expertise and resources, surveillance systems detecting unintended consequences early, and international frameworks for coordination. It also includes research into risks themselves, studies exploring how gene drive spread, how edited genes might have off-target effects, how AI predictions can fail. Equity must be a core commitment, not an afterthought. Benefits should be accessible to all who need them, regardless of geography, wealth, or political power. This requires addressing structural inequalities shaping who benefits from medical innovation. Equity is not only a moral imperative but a practical necessity. Infectious diseases do not respect borders. Public engagement and education are essential. Building public understanding requires dialogue, where scientists listen as well as speak, where public concerns are taken seriously, and where values are negotiated rather than dictated. It requires transparency about uncertainties and risks. And it requires trust, built through consistent, honest communication. We must also think seriously about dual-use risks. The same tools that heal can harm. There are no perfect solutions, but there are measures that can help: monitoring synthesis of dangerous sequences, restricting access to sensitive information, fostering a culture of responsibility, strengthening international agreements, and investing in attribution and response capabilities. At a deeper level, moving forward responsibly means cultivating certain virtues. Humility, recognition that we do not know everything. Caution, willingness to slow down when risks are high. Courage, willingness to act despite uncertainty. Compassion, keeping human suffering at the center. Justice, ensuring benefits and burdens are distributed fairly. And foresight, thinking across generations. These virtues are cultivated through education, through practice, through institutional structures that reward them. They require creating incentives for responsible behavior, celebrating scientists and institutions prioritizing safety and equity, and holding accountable those who ignore warnings. We should also remember that disease has been part of the human story, shaping culture, memory, and meaning. As we work to eliminate disease, we must be intentional about preserving lessons and insights that struggle has produced. This means teaching history, remembering those who suffered, and finding new ways to cultivate empathy, resilience, and solidarity. There is also the question of what we aspire to. Is the goal simply to eliminate disease, extend lifespan, enhance capabilities? Or is it something broader, to create a world where people can flourish, where communities are resilient and ecosystems are healthy? A truly wise approach would situate biotechnology within a larger vision of human flourishing, using it as one tool among many. The thought experiment we have conducted is useful not because we can change the past, but because it clarifies what is at stake in the present and future. It shows that technical capability is necessary but not sufficient, that every intervention has consequences, and that the wisest path combines ambition with caution, innovation with oversight, and power with humility. As we close, let us hold several truths together. Biotechnology is powerful and holds immense promise for reducing suffering and extending life. It is also dangerous, capable of causing harm through misuse, accident, or unintended consequences. It is rapidly advancing, and it is unevenly distributed. Navigating this landscape requires all of us to engage thoughtfully, to ask hard questions, to demand accountability, and to insist that power to reshape biology be wielded with care. The diseases that once seemed invincible have shaped who we are. They have left scars in our genomes, in our cultures, in our collective memory. We now have tools that might have prevented them, or that could prevent the next generation of diseases. Whether we use those tools wisely depends not on the science alone, but on the values we bring, the structures we build, and the choices we make. The story of disease is not over. It is transforming, taking new forms, presenting new challenges. But the story of human response continues as well. We are participants in it, whether we choose to be or not. The question is whether we participate thoughtfully, with eyes open to complexity and hearts committed to the common good, or whether we stumble forward, dazzled by capability and blind to consequence. The night is quiet. The room is soft. The questions remain, but they are questions we can live with, questions that sharpen rather than paralyze, that invite curiosity rather than fear. We have imagined much: diseases prevented, lives saved, systems transformed. We have also acknowledged much: risks, trade-offs, uncertainties, losses. What we carry forward is not a blueprint but a framework, a way of thinking about power and responsibility, about what it means to reshape the biological world and our place within it. Rest now, with these thoughts settling. Tomorrow, and all the tomorrows after, the work continues. The decisions will be made, the tools will be deployed, the consequences will unfold. We cannot know the future, but we can approach it with care, with courage, and with quiet determination to leave a world that is, if not perfect, at least a little better, a little wiser, a little more just. That is enough. That is the work. That is the responsibility of possibility. Sleep well.